group in front of your name. Okay, before I also start, I will like to explain the mechanism. First, I would like to start with group 8 till group 14. So here I would give the time to group 8, consists of the member of group 8 is Miss Widya Rahma, Miss Febri Agnes, Mr. Slamet Rianto, and only 3 people, 3 persons. Okay, please, group 8, time is yours.
Okay, thank you, sir. First, I would like to invite my friends to do the share screen.
Presentation started by introducing your member of group. Okay.
Okay. Okay, good morning, everyone. We are from group 8. The members of our group is Mr. Slamet Rianto from IPB University, Febri Agnes from IPB University, and myself, Widya Rahma from IPB University. Today, we will present our discussion about our final project, the protein-protein interaction in hypertension. Next. So in our final project, we are presenting and analyzing the interaction between the protein-protein in hypertension, and the materials and methods that we use in this final project is the protein database that we are get from the list of proteins in TXT format file. Then the protein database from the file we are processing with the string web server, or the search tool for the retrieval of interaction gains or protein. This strings web server is a biological database and web resource of known and predicted protein-protein interaction. Then from the string, we do the processing file with Cytoscape, the open source bioinformatics software platform for visualization.
Ms. Vidya, can you open your video before I continue the presentation?
Sorry, maybe I cannot open my camera because when I open camera, my network is not stable.
Okay.
Okay.
Please continue. Yes.
Cytoscape, an open source bioinformatics software platform for visualizing molecular interaction networks and integrating them with gene expression profile and other state data. Then from the Cytoscape, we do the programming code with Google Colab, then analyze the finalize the result to Microsoft Excel. So this is the string web server website, the interface of the website. Here are the multiple proteins that we are get. And then from the string website, we conclude this interaction, the picture of the interaction of the hypertension protein. This is that we can see here so many interaction of the protein from the hypertension. And next, from that network, we are processing them with the Cytoscape. And this is the clustering from the Cytoscape. Then we do the clustering to choose the important protein. Next slide. So here the processing of the important protein, and from the cluster, we are choose the lowest p-value. It's about the first node, the lowest nodes. And after that, the next slide. And here are the protein that we can conclude after the protein clustering. So from this step, we can choose the parameter between the centrality, the closeness, and the stress. And after that, the output file, we are processing it with the Google Collaboratory. Next slide. So here, first we are importing the Python resource code, and then we do some data finalization. And here the overall result from the cytoscape. After that, next slide. After that, we are do the parameterization with the Excel, and we get the highest value from the overall centrality value, the SCN1B gen. And the SCN1B gen is the protein that have the significant value to do the hypertension. The next explanation will be delivered by my friends Febri.
And the protein that has an important role related to hypertension is SCNN1B. The amyloid-sensitive sodium channel beta subunit, or SCNN1B gene, encodes the beta subunit of the epithelial sodium channel, which is involved in blood pressure homeostasis due to Zhong et al. 2016. Sorry. Sorry, can anyone still see my screen?
Yes.
Which slide, Mr.?
Protein that has an important role as SCNN.
Oh, it's not working. Seems like it's not working. Maybe I can stop share first and then.
Yes. Start again. Start share screen.
I will continue. And this is the information about SCNN1B. The gene ID is 6338 with official symbol is SCNN1B and official full name is sodium channel epithelial 1 subunit beta, and the gene type is protein coding organism in Homo sapiens and also known as BESC1, ENAC-B, and etc. And mutations in this gene have been associated with pseudohypoaldosteronism type 1 or PHI1 and Little syndrome. Expression in esophagus, colon, and 11 other tissues. Next is the SCNN1B gene provides instructions for making one piece, the beta subunit of protein complex called the epithelial sodium channel or ENaC. The ENaC plays a critical role in body fluid volume and Na plus homeostasis, which underlie the pathogenesis of salt-sensitive hypertension due to Green et al. 19 and 90. And this is the 3D structure of ENaC. Next is health conditions related to genetic changes of SCNN1B. It is called Little syndrome. People with Little syndrome have high blood pressure known as hypertension and low levels of potassium in their blood known as hypokalemia. And the mutations in the SCNN1B gene associated with Little syndrome. And as a result of the mutations, the protein is not degraded and more inert channels remain at the cell surface. The increase in channels in the surface cell abnormally increase the reabsorption of sodium followed by water, which leads to the hypertension. And reabsorption of sodium into the blood is linked with removal potassium from the blood, so excess sodium reabsorption leads to hypokalemia. And high blood pressure is the leading cause of morbidity and mortality due to cardiovascular-related disease such as stroke, heart failure, myocardial infarction, and chronic kidney disease, due to Hall et al. 2016. And these are the references that we used. And I think that's all for Group 8 presentation. Thank you all for paying attention.
Sorry, sir. I think you still mute your microphone.
Okay, thank you, the Group 8. I would like to ask one question, and I prefer Mr. Slamet will answer this question, okay? Because Mr. Slamet do not present in this presentation. So, what actually the advantage and the disadvantage using protein-protein interactions in this problem? Is it clear what the advantage and disadvantage?
Okay, thank you, Mr. Wisnu. Sorry, I outside in room because the vaccine. Sorry.
Okay.
Yes. My knowledge maybe advanced protein network, because we know to can be analyzed about protein that's related some disease, and then we based on the result of analyze, we can to prefer about to some disease, and but sorry, because the in the my experience about the chemical, maybe I little know about this about the protein. Let just my question, my answer, correct me if I wrong. Thank you.
Is there any comment or answer from the other members? For example, from Ms. Widya and Febri? Hello?
I think that's enough, sir.
Enough? Okay. Okay. Thank you, Group 8. I think it's enough, and I will call the Group 9 to continue the presentation. Thank you.
Thank you very much, sir.
Yes.
You're welcome.
So
Thank you.
close the share screen. Okay. Okay, Group 8. Please, you have 10 minutes, maybe 8 minutes for presentation, and then 2 minutes question answer. Please, Mr. Eko, Ms. Ida Ayu, and Ms. or Mr. Kimberly. Oh, Ms. Kimberly from UPM.
Yes, hello, good morning.
Okay. Please, first introduce your member of group.
Okay. Is my slides available?
Yes, available.
All right. So a very good morning to everybody, and a very good morning to the committee members who have successfully run this program, and a good morning to all the participants. My name is Kimberly. I'm from Group 9, and I'd like to introduce my teammates, Ms. Idayu, Mr. Eko, Ms. Fani, Prof. Hilda, and also
All members attend this presentation.
the three of us will represent the whole group.
Huh?
The three of us, me, Mr. Eko, and Ms. Idayu will be representing.
Okay.
Yes. So let me start off. This is our sort of table of content. I will be presenting what is diabetes, and my teammates will be presenting the analysis of the PPI network. So to start off, what is diabetes? It's a condition in which the body does not properly process food for the use of our energy, and most of the food we eat is turned into glucose to use for energy. The pancreas is where the hormone called insulin helps glucose to get into the cells of our bodies. We will be talking about type 2 diabetes, whereby it is an impairment in the body. It is an impairment in the way the body regulates and uses sugar as fuel, and the long-term condition results in too much sugar circulating in the bloodstream. Therefore, eventually, high blood sugar levels can lead to the disorders of the circulatory, nervous, and immune system. You can see here the picture on the right where it is 4.8. This is a small machine that will tell you that will show the blood glucose levels in your body at a certain point. And this is a healthy level, 4.8. A high level would indicate number 6 and above. So the first step in the process of finding the right drug for a disease is to identify the significant proteins associated with the disease. And the problem here is the expensive and long experimentals lead to a computational approach that is considered more efficient, which is the solution to the problem. And this computational approach is to identify protein significant association with a disease, and is proposed as a solution to experimental constraints. And one of them is by constructing and analyzing the topology of the protein-protein interaction network. I'll pass this time to Ms. Idayu.
Okay, thank you. The PPI information data were imported into Cytoscapes, an open source software platform, and a PPI network was easily constructed. Clustering with overlap neighborhood expansion, such as cluster 1, was used in Cytoscape network in order to discover densely connected regions in the network with possible overlap. The quality function can guide it by cluster 1 by growing dense region out of small seeds. The parameter that we choose is minimal density is 0.05 and the degree is 6. Next slide.
Sorry, Mr. Idayu. Is it this slide or you prefer the previous?
Okay, this slide. This is the calculated. Okay, based on data calculation using Python, UBB protein has the highest PCCC degree stress and overall value. This indicates that the UBB protein is a significant protein. Next slide. UBB or ubiquitin has a major role in targeting cellular protein for degradation by the 26 proteasomes. It is also involved in the maintenance of chromatin structure, the regulation of gene expression, and the stress response. Ubiquitin is synthesized as a precursor protein consisting of either polyubiquitin chains. Another research say that type 2 diabetes has correlated with increased ubiquitin levels. This is the 3D model of UBB protein. Next slide. The ubiquitin proteasome system, or UPS, is the major pathway for intracellular protein degradation in most organs, including the heart. UPS controls many fundamental biological processes such as cell cycle, cell division, immune response, antigen presentation, apoptosis, and cell signaling due to Sukla and Ravik 2020. Next slide. Okay, recent study confirmed that UBB is an important marker for cardiac remodeling and a potential target to modulate UPS-mediated cardiac remodeling. Diabetic cardiomyopathy, or DCM, is a disorder of the heart independence of hypertension and coronary arterial disease. The condition results in wide range of structural abnormality to the myocardium and eventually leads to left ventricular hypertrophy, or LVH, diastolic and systolic dysfunction, or a combination of all three, by Shukla and Rafiq on 2020. The prevalence of diabetic cardiomyopathy is increasing in parallel with the increase in diabetes mellitus. Diabetic cardiomyopathy is initially characterized by myocardial fibrosis, dysfunctional remodeling, and associated diastolic dysfunction, later by systolic dysfunction, and eventually by the clinical heart failure. Next slide. Diabetes for ubiquitin proteasome mediated degradation. From the studies of transgenic animal models and humans, there is an evidence that inappropriate level of signaling molecule strongly affect insulin action. It appears that controlled degradation of specific protein by the ubiquitin proteasome system plays an important role in the execution of various biological functions, including insulin signaling. Insulin promotes cellular growth and maintenance by a wide variety of both anabolic and anticatabolic action, including the inhibition of five. So this is the illustration of insulin signaling molecule under proteasomal chart. On the left side, normal insulin signaling pathway leading to balanced metabolic and metagenic action. On the right side, SOCS protein distal to the IR mediating ubiquitin proteasomal degradation of IRS. For the next slide, Mr. Eko will continue my presentation.
Thank you, Ms. Ida. Because the ubiquination process affects the regulation of insulin signaling, cascade, and insulin action, it appears that there may be several hidden drugs targets for diabetes and its complication in the ubiquitin proteasome system. Considering the large number of the transcription factors and UB ligases present in the cell, it is possible that UB or UB-like mediate regulation or transcriptor factor localization will participate in the protagonist of diabetes and its complication. However, the recent experimental evidence strongly suggests that proteasome inhibitors may indeed be beneficial in certain pathologies, such as in cancer, asthma, brain infarct, and autoimmune encephalomyelitis. There may be a feature in bioprospecting of natural products, curcumin, an active component of Indian spice, turmeric, has been shown to impair ubiquitin proteasome system. A different approach to drug development can be the development of small molecules that bind and inhibit specific E3S. Next slide, Miss. The conclusion is UBB protein is a significant protein in diabetes based on the PPI network. This is proven by the research that UBB or ubiquitin has a major role in diabetes. Ubiquitin has role to targeting cellular protein for degradation by the 26 proteasome. Ubiquitin also an important marker for cardiac remodeling and potential target to modulate UPS-mediated cardiac remodeling. ubiquination process are shown to affect the regulation of insulin signaling cascade and insulin action. It appears that there may be several hidden drug targets for diabetes and its complication in the ubiquitin proteasome system. Okay, that's our presentation now. Thank you very much.
Okay, thank you, Group 9. Okay, I have two questions. First, I hope the other members will answer this question. Yes. First is it the PPI analysis using cytoscape and also topological approach or clustering is useful for understanding the biological process related to the specific disease? This is the first question. And the second question is, what the most difficult to implement this method? Please, maybe the other members can answer this question. But if there is no other members, so I hope the other members that not present in this presentation should
Thank you.
answer the question.
Thank you very much for the question. Maybe I will try to answer the question.
Okay, Bu Revia.
We think this method is very useful, and then to apply about these methods, maybe it's very difficult for the person who haven't know about the program and the others. But for the person who has known that, it's very easy, maybe.
Okay.
I think that's all. Thank you very much.
But which part from the method is most difficult?
I think for to put our data and to use the program PPI to find the interaction of the protein-protein interaction. So we want to know how the protein-protein interaction for one disease like protein like that. And for the person who haven't know about how to use the program, maybe it's quite difficult. Okay. Thank you very much.
Yes, thank you very much, Group 9. So I think it's enough. Then I would like to call the Group 10.
Thank you very much, Mr.
Thank you.
Welcome.
Group 10, Mr. Bayu Andika, Miss Agnes.
Yes, sir. Let me share our PPT first.
Mr. Iwan, okay. There's 6 members.
Yes, but the one who are going to presentate is me, Agnes, and Iwan Jeffrey. Sorry.
Why become six members? Why?
Before that, the group 12 is just
12,
one person.
okay. Only Mr. Mayu, yeah.
Yes, Miss Mayu, but
Miss Mayu, sorry.
There's her friends that would join the final project. So we make a collaboration, the group 10 and 12.
Oh, 10 and 12, yeah. No.
Yes.
Okay. Okay.
So I think Iwan would start the presentation.
Good morning, Mr. Wisnu Ananta, and good morning, everyone. Today we form Group 10 and 12 Regression Final Project, Protein-Protein Interaction Analysis for Alzheimer Disease. Is this member of Group 10 and 12, and introduce me, Iwan Jeffrey from IPB, and my friend Argus Giovanni from ITB will represent our group. There are several sections that we will explain. First, introduction, second, data result, three, discussion, and fourth, conclusion. Introduction. The meaning of Alzheimer's disease is characterized by neurodegenerative mental loss and
Sorry,
of
Mr.
mental
Iwan, maybe
capacity. The development of
it's better. Hello, it's better if you close the camera, maybe because the voice is not so clear. Okay.
Okay. Alzheimer's disease is characterized by a neurodegenerative dementia including disorder that result in a progressive and ultimate fatal loss of mental capacity. The development of AD is accompanied by the granular spread of sticky flux and clump of tangled fiber that disrupt the delicate organization of nervous cells in the brain, undermining the normal communication among brain cells. Why have Alzheimer's disease? Alzheimer's disease shows the importance of both amyloid beta, plaque, and tau protein neurofibrillary tangles in what he called the disease of forgetfulness. These two proteins are very different. A beta form extracellular aggregate and plaque, whereas tau protein are intracellular protein involved in the stabilization of action by cross-linking microtubules can form disorganized tangles. Next, protein-protein interaction. Protein-protein interaction are physical contact of high specificity and stabilization between two or more protein molecules as a result of biochemical events stirred by interaction that include electrostatic force, hydrobonding, and the hydrophobic effect. Many are physical contact with molecular association between chains that occur in a cell or in a living organism in a specific biomolecular content. Protein-protein interaction are associated with A activities and pathway in the cell and in the regard are promising target for drug discovery. This is data result. We have data result using cytoscape and cytocluster. Next, cluster 1 algorithm. Analysis network parameter between centrality, closeness, centrality, stress, degree, and overall centrality. We have 16 node and p-value 0.008 mean the cluster is significant. The higher the result of each parameter means that the protein is more relevant to disease. The result of analysis parameters sorted by value of overall centrality have two highest relevant is FGA and Sirvina1. Two protein is significant overall value than other protein. High relevant is P for each B and A for E. The two protein is second significant than other protein.
Okay, so next we're going to talk about the discussion that according to the data result that have been got before. So from the data result, we got that these four proteins are the most relevant proteins according to the Alzheimer's disease. And now we're going to prove how is the protein is related to the disease. Are the proteins really significant to the Alzheimer's disease? So we conduct study literature to know how this protein can relate to the disease and what is the role of the protein related to the Alzheimer's disease. And the first one is FGA, or fibrinogen alpha chain. And it is stated that the accumulation of FGA was enhanced in Alzheimer's disease patient brains. And now that the FGA also triggers reactive astrogliosis, inhibits neurite outgrowth, increase vessel wall inflammation, and vascular permeability in endothelial and smooth muscle. And so this is contribute to the progress of Alzheimer's disease. And the second one, the SERPINA1. The SERPINA1 is a gene that encode the protein, and the protein can have a modification which is the sialization modification. And the sialization modification is a protein abnormal post-translational in the phosphorylation and also glycosylation, which we know that this process is related through the glycolysis mechanism, and this is recognized as a pathogenic mechanism in neurodegenerative disorders. And also, SERPINA1 can form a misfolded and self-aggregate bronze structure, and then it is also an important modulator of neuroinflammation. And yes, these two proteins is the highest relevance proteins according to our data result. And the other two proteins has a high number of overall centrality, and so we also conduct study literature to know how these proteins can affect the disease. And yes, there is a role of the protein that is related to the Alzheimer disease. The P4HB is related through the MAM hypothesis. So we know that the amyloid beta is the cause of the Alzheimer disease, as has been mentioned before. The amyloid beta is generated or is produced in the endoplasmic reticulum or ER. And then because of that, we know that it is physically and biochemically connected to the mitochondria. So it is called mitochondria-associated ER membranes or MAM. And this P4HB gene encodes protein PDIA1, which can restrain protein in ER. And this is related to the MAM mechanism. And the last one is the ApoE. Human express 3 genetic of ApoE variants, and it can differentially affect Alzheimer's disease. And in brain, ApoE containing lipoproteins are secreted by activated astrocytes and microglia. So as we mentioned before, the cause of the Alzheimer's disease is two. It is the amyloid beta and also the tau protein. And later we will discuss how can this role affecting the cause of the Alzheimer's disease further. So this is the explanation for the fibrinogen alpha chain. Yes, in this picture, you can see there is HCAA. HCAA is hereditary cerebral amyloid angiopathy, which is a pathological feature of Alzheimer's disease. And as I have mentioned before, amyloid beta is the cause of Alzheimer's disease, but it doesn't act alone. It needs a forming agent. So the forming agent one that can make a complex is the blood clotting protein fibrinogen. Yeah, so the amyloid beta here and the fibrinogen will form a complex together, and then the result from these deposits around the cerebral blood vessels is the disruption of brain vasculature, which leads to the pathological feature of Alzheimer's disease. Next, it is the SERPINA1. The SERPINA1 can form an aggregation to form polymeric filaments. And this is occur if there is a space available for beta strand insertion, as is the main beta sheet of the conformationally unstable variants of alpha-1 antitrypsin. Alpha-1 antitrypsin is the other name of SERPINA1. And these beta linkages are relatively non-specific, and as I mentioned before, can potentially occur wherever there is a free peptide sequence, and because of the insertion. And this is what we call beta amyloid peptide of Alzheimer's disease. So next is the P4HB. Yes, this is related to the MAM hypothesis, and MAM is an extracellular lipid structure that has so many roles in cholesterol and phospholipid lipid metabolism, calcium homeostasis, and in mitochondrial function and dynamics. And this P4HB gene encodes protein disulfide isomerase A1 that can restrain protein in ER related to the MAM mechanism. So this is the mechanism causing Alzheimer's disease through the increased MAM-mediated and ER mitochondrial communication, and P4HB gene and the protein is affecting in this way. Yes, and the result is Alzheimer's disease. So I think it is a bit difficult to understand if we just read the text. And the last protein is about ApoE, and the ApoE mainly influence the pathogenetic of Alzheimer's disease by affecting amyloid beta pathology and can also trigger neuroinflammation through this pathway. Yes, this is the amyloid beta, the tau pathology, and the ApoE can affect and resulting in Alzheimer's disease. And yes, it is a bit difficult to understand if we just read the text. So this is the big picture of the Alzheimer's disease pathway and how can these 4 significant proteins effect in the pathway of the Alzheimer's disease. So the first one is FGA. This FGA, as we mentioned before, is the alpha beta interacting partners that can interact with amyloid beta and form the toxic alpha beta. And toxic alpha beta here is the cause of Alzheimer's disease. And the second one is SERPINA1. SERPINA1 is affecting, because of its serialization modification, it's affecting the phosphorylation and glycosylations, which is affecting the glycolysis pathway. And glycolysis pathway is also contribute to the Alzheimer's disease. And other than that, SERPINA1 is also the modulator of inflammation. So it affecting in here, this neuroinflammation can make the Alzheimer's disease. Then the third one is P4HB. P4HB through the MAM hypothesis, can also affecting the amyloid beta here because the amyloid beta is regulated by the mitochondria. And the amyloid beta can form toxic amyloid beta and then causing the Alzheimer's disease. And the last one is the ApoE. ApoE here is the ApoE is a non-enzymatic alpha beta clearance, and this can impact cholesterol, lipid, impair vesicle endocytosis, and also this result can lead through the Alzheimer's disease. So this is the proof that our 4 proteins, our 4 major proteins are affecting Alzheimer's disease pathway. Okay, next, we're searching for the main compound for treating the Alzheimer's disease. And we've got from the IJAH analytics that there are 5 components that can be used as the treatment for Alzheimer's disease. The first one is stigmasterol, and we conduct also the literature study to know its role. And the stigmasterol can directly decreasing beta secretase activity and reduce the gamma secretase components, reduce cholesterol and presenilin distribution, and decrease the ACE1 internalization, which can also seen in the Alzheimer's disease pathway. And next is psimane that have antioxidant and anti-inflammatory properties and also the antifebrile effect. And the third one is nerol, which has also antioxidant activities and also the acetylcholine esterase inhibitory activity. The fourth one is linalool, which has anti-inflammatory effects. And also the last one is terpinane-4-ol, has an antioxidant properties to neutralize reactive oxygen species that can lead to Alzheimer's disease. And so these compounds can be affecting the Alzheimer's disease pathway through this one. So from the first one is the stigmasterol. For I said before that the stigmasterol is reducing the BACE1 and also reducing the gamma secretase so that the amyloid beta will not be formed and the Alzheimer's disease can be prevented or can be treated. Then from the oxidative stress can be against by antioxidant activities, and the one who has antioxidant activities is psimane, nerol, and terpinane-4-ol. So it can inhibit the oxidative stress, which can lead also to the Alzheimer's disease. And also there is this neuroinflammation, as I have been said before also, the component has anti-inflammatory activities. So psimane linalool, which has anti-inflammatory activities, can inhibit the neuroinflammation, which can prevent the Alzheimer's disease. And the last one is nerol. Nerol, other than have antioxidant activities, it also has an acetylcholine esterase inhibitory activities, which can prevent the synaptic dysfunction, synapse loss, cognitive, and decline. And for your information, acetylcholine esterase inhibitory is the popular one that we use as a conventional drug to treat the Alzheimer disease.
Sorry, Miss, maybe time is almost up.
Oh,
so
okay,
please.
yes, just a little bit. This is the plant that contain the compounds, the Pterocarpus santalinus, Viper arboreum, and also Artemisia capillaris. And so this is our conclusion that using the analysis, we can show that FGA, SERPINA1, P4, HB, and ApoA are the proteins with high relevance to the Alzheimer disease. And those proteins are proved to have effects in the AD pathway. So we can see it in slide 16 before. And those proteins have increased numbers in AD patients. So we can use it as our drug targets for our developing drugs and also as the biomarkers. And the analyzing plant compounds related to the AD using IJA analytics shows that stigmasterol, picimine, nerol, linalool, and terpinol can be used as treatment for AD by affecting AD pathway in the slide 18. And so these are our references that we use for our final project. Thank you for your attention. Sorry for the late of the time.
Okay, thank you. Group 10 and 12, yes, it's very interesting because I'm surprised because your group also use IJA analytics. So it's very interesting. So there's my question: how you use IJA analytics? For example, if you already have information about protein, then how to use the IJA analytics for analyzing or for finding the compounds and also plants? Can you explain? How? Maybe the other members can answer the question. For example, here is Mr. Bayu or Miss Rani or Miss Mayu or Miss Nelly. Is it clear my question? After you've obtained the protein, then how can you connect, or how can you use the protein to find the compound or plant by using the IJA analytics? Or maybe the other members, please answer the question. My question is very simple, I think, because you already done. Maybe Miss Agnes or Mr. Iwan can answer the question.
We are use IJA analytic because I from my university and I magang in tropical biofarmaka of this IJA analytic. Terus to use IJA analytic, we can open a website and we can search homim ID of disease, and we can search and we search uniform ID to protein.
I mean, your input for IJA is disease or protein that you find from cytoscape analysis or PPI analysis.
The first I search homim ID, the disease, but I can.
So your input is homim. Your input is.
I can find both disease. Kemudian next I input protein to IJA analytic.
Okay. So you put protein that obtained from the PPA analysis to IJA analytics.
Yes.
Okay. Okay. Thank you for group 10 and 12. I think it's enough. Then I would like to give the time to group 11.
Yes,
Mr.
sir.
Aziz. Okay, Miss
Would you please
Kiani. Okay.
let me share the screen?
Yes.
Okay.
Okay.
Is it shows?
Yes.
Okay, thank you. Good morning, konnichiwa, wilujeng enjing, assalamualaikum warahmatullahi wabarakatuh. Imagine the day without technology. Maybe today we haven't discovered what is happening inside our body or what is happening inside our cells within just a minute or maybe just some seconds. Is it interesting, right? Good morning, ladies and gentlemen. We are from Group 11. Our member is Aziz Riyadi from IPB University, Zahir Torik from University of Indonesia, Vinda Maharani from Bandung Islamic University, Maki Suzuki from Gifu University, and me, Elgiani Yasifa from IPB University. In this opportunity, we would like to talk about the analysis of protein-protein interaction on osteoporosis diseases. Aging population is a trend of 21st century. And the aging population is an increasing older people population because of declining fertility rates and rising the life expectancy. According to World Health Organization, the population of people aged 60 and older is about 900 million people in 2015, and it is predicted to double to 2 billion people in 2050. Is it a lot of amount, right? But the aging population is happen globally and followed by many opportunities, but also several public health challenges that we need to prepare for. One of the biggest challenges is osteoporosis. Osteoporosis can lead to reduce the quality of life and even death. And according to International Osteoporosis Foundation, there are 200 million people that are suffering from osteoporosis. 1 in 3 women and 1 in 5 men over the age of 50 years will experience osteoporosis fracture in their lifetime. And it's quite bad. But what is osteoporosis? My friend Aziz will explain about it.
Osteoporosis is a condition where the low bone density and microarchitectural deterioration which can predispose to rigidity fracture. The pathophysiology of osteoporosis is yet unclear. The potential mechanism underlying osteoporosis pathogenesis involves an imbalance between bone resorption and formation. Tumor sources factor TNF receptor-associated factor 3 or TREF3 and FKB signaling pathway, node signaling pathway, WNT or beta-catenin pathway, and phosphatidylinosytol 3 kinase or PL3K or protein kinase B, AKT, PL3K or AKT pathway. Therefore, osteoporosis is also known as the silent disease. In recent years, osteoporosis and its complications including deformity, disability, and depression have been a major public health concern among aging population worldwide. Race, age, sex, other bone metabolic disease, endocrine system disease, rheumatic immune disease, age, drugs, and lifestyle contribute to osteoporosis pathogenesis. The cost of nursing and medical care for osteoporosis individuals is enormous, imposing a heavy burden on the family and society. Next. We use bioinformatic method to construct a protein-protein interaction network and analyze the functional models in the network, providing new view of osteoporosis and evidence for the need for further osteoporosis research.
Many biological functions involve the formation of protein-protein complex, like proteins are executor of the genetic program and rarely act alone because they have to interact with each other to realize the complicated biological processes in all living creatures, including human beings. Next. Yeah, there are several stages for application of PPI analysis. You can find lists of protein that you want for the interaction, maybe in string database or NCBI website. And for the prediction of interactions protein, you can use cytoscapes and continue exercise like pretty much a 5 cluster or protein analysis. After clusters are formed in cluster 1, the formation of new cluster are followed by new seed nodes. Next. Yeah, network-based systems biology approach have emerged as powerful tools for analysis of molecular mechanism of disease. One of them is osteoporosis. Previous studies have shown that primary osteoporosis is associated with genes mutations. So based on this research, we want to explore the functional modules of the PPI network and the related pathway participating in primary osteoporosis.
Now we will discuss the step we took to build a protein interaction network from data on protein that play a role in osteoporosis disease. In conducting this experiment, we used bioinformatic method to build a protein interaction network and analyze the functional modules in the network. Next. First of all, before we build a protein interaction network, we must have a data list of proteins that have an effect on osteoporosis disease. We can get this data through the UniPrecord website. UniPrecord is a website which provides a comprehensive database of protein sequence and annotation data. But for this experiment, the list of protein data used is available on the OCW link, so we just need to download it. After we have the data for the protein, we have to enter the list of protein into the string website to get a predictive picture of a protein interaction. String is database of known and predicted protein-protein interaction. On the website string, we select multiple protein and copy-paste the list of protein associated with osteoporosis disease from OCW. Then select Homo sapiens as the organism. Next, in the advanced settings, we select the highest confidence for the required score. After getting the result, then save the result by clicking the download as simple table text output. You can access the UniPrecord website at the link www.uniport.org, and you can access the string website at the link string-db.org. Next. After that, we built a visualized PPI network using Cytoscape software. And Cytoscape software is an open source software platform to visualize complex network and integrate them with all kinds of attribute data. In Cytoscape, we use Cluster 1, which is a plugin for Cytoscape, which contains several clustering algorithms. Cluster 1 is basically looking for group with high cohesiveness. Before we use cluster 1, we first change the setting to be like below. From this Cytoscape application, we can get data about betweenness centrality, closeness centrality, degree, and stress data from all protein contained in the selected cluster. Next. After we get the data, we need to calculating the overall centrality using Python OR. A function is a reusable block of programming statement designed to perform a certain task. To define a function, Python provides the def keyword. Overall centrality which Python can be compiled using IDE, Capitale, Anaconda. Other IDE also can be used with some improvement. By using Python and R, we can get the final data which we will later use to draw conclusion from this experiment. Next, for the result will be presented by Suzuki-san.
Okay, I'll explain the result and discussion. This is a result of analyzing by string. We can see inflammatory cytokines such as TNF, IL, NF-kappa B, and other proteins considered to be related to osteoclastogenesis, such as TRAF6, CTSK. Next. And this is a result of a cluster which has a lowest p-value. As the table shows, IFIH1 is the most important protein for osteoporosis. Next. Here's a discussion. Osteoporosis is mainly caused by excess of osteoclastogenesis, and TRAF6 and NF-kappa B are strongly related to osteoclastogenesis. The result has these proteins, but the best is IFIH1. IFIH1 is interferon induced with helicase C domain 1, and it encodes melanoma differentiation associated protein 5, MDA5. MDA5 is an intracellular sensor of viral RNA that triggers the innate immune response and plays an important role in enhancing natural killer cell function. So we guess that osteoporosis is more related to other immune system than osteoclastogenesis. Next. That's all for Group 11 presentation. Thank you for your attention.
Okay, thank you. For Group 10, I just want one question. Do you think that the PPI analysis using cytoscape and also topology-based approach, for example, by calculating the overall centrality, degree centrality, and so on, etc., can be used for example, to find the biomarker for specific or certain disease? Is it clear, my question? Hello? Please, maybe anyone can answer.
Thank you for the question. I think I would like to try to answer the question. I think these programs or website is also can use to identify or find the biomarker because it has the same function that we need to filtering what is the best biomarker, and then the result will shows by also the best biomarker. Maybe the other members would like to add the answer.
Okay. Enough?
Or maybe that's enough for us. Thank you.
Okay, thank you, Group 11. I think it's interesting presentation. Okay, so thank you. I would like to call to give a time to Group 13, yes, because 12 is already present. Already joined with Group 10. So I ask group 13, please, Mr.
Sorry, Mr.
Yes.
Wisnanta.
Yeah.
May I get an access to share?
Oh, you cannot share your screen? Huh?
Oh, yeah. Okay. Thank you. Wait. My screen is get display on your screen, Mr. Wisnanta.
Okay. Yes. Okay.
Okay, I'm the first one to presentate in group 13. And good morning, everyone. We are from group 13, consists of me, Maheswari Alfira Dwicasaria, Arif Rahman Hakim, Muhammad Riki Damara, and Irano Vitasari. Today, we want to present our group result of protein-protein interaction analysis with arthritis protein. Arthritis with the common form rheumatoid arthritis is one of inflammatory disease that the immune cells are attacking healthy joints. Arthritis have some symptoms that mostly happens in bones and muscles such as joint pain, joint swelling, osteoporosis, and muscle wasting. According to Swallows and Bush 2015, arthritis is a systemic chronic autoimmune disease that makes inflammation of connective tissue, especially on joints. Therefore, its cause clinical description is pain, stiffness, unmoving, and swollen on joint. Next. Next. Protein-protein interaction plays key role in predicting the protein function of the target protein and drug ability of the molecule. The procedure of the protein-protein interaction analysis are divided into parts using some application and web server such as UniProt, String, Cytoscape, Cytocluster, and JupyterLab. First of all, we have to search the protein in UniProt database. The one we should choose is Homo sapiens. Next. Yes. When the result is come out, we have to download the entire protein with the Excel format. And next. We have total 258 proteins that have role in arthritis. And then the data is input into the String web server. And then we choose the higher confidence and click search. The presentation, yeah.
Next subject is
Ricky.
Thank you. Next subject is construct the protein-protein interaction and clustering the protein interaction. After we click search, this is the result protein analysis obtained. After that, click continue to the next process. And then seen in the picture of protein-protein interaction in arthritis. Also seen the middle, which is the strongest interaction. Next. And then download the data in TSV format. Next. Next step is input the downloaded in string data into Cytoscape. Next. And then select the first and the second column. Next. Run the Cytocluster app in Cytoscape and choose cluster 1. Next. Next subject is analyze the cluster and calculate the overall centrality from every protein on the cluster using JupyterLab. Next. In the picture, we change the basic and range parameters and we get to cluster. Seen the right picture about to cluster. Next. Next, analyze to calculate some parameters. Next. After that, seen the result of parameters in the table, and then the left column escape, witness, centrality, closeness centrality, degree, and stress. Next. And then export the table into CSV format and then coding the result in Jupyter Lab app. We get the final result and present it Excel format.
Start from this, I'll continue the presentation. Okay, the parameter which we use to determine this result is first local measurement. From last slide, we know that the four parameters we take is between centrality, closeness centrality, degree, and stress. But what is the meaning of those parameters? Okay, the first one is the degree. Degree is degree of node is how many connection or edge it has. So when we see the cytoscape result, or maybe the string result, like this one, this node degree, it means how many connection that this one node have. So this parameter is called local. And from and the another one, relative measurement of all nodes in the network. First one is closeness. By calculating the average distance or shortest path from one node to all nodes in network. Closer the nodes to all nodes, it will make this protein become more significant. And then the betweenness. The betweenness of a node is the number of the shortest paths through that node. The more number of shortest paths that pass through the node, the more significant this protein. And the last one is stress. Stress is just total of shortest path that pass the node. Okay. From closeness, centrality, between centrality and stress, it looks like very similar, right? So I'll explain the differences because just a little thing differences with these three parameters. Okay. For the first one is closeness. You could see the node in the middle, right? So because this node is close with another node, so this node or this protein become more significant. And then the betweenness. The betweenness is the shortest path which pass through the node. So you can see this node, yeah. You can see the edge or the line which is passing through this node is so many, and you can see that there is many line from many nodes or different nodes. So because another node, their edge is passing through this protein, so this protein is become significant. It's called betweenness centrality. And for the stress, is edge or line which is on the around this node. You can see the line around this node is so many, so this protein become more significant. That was the explanation why we pick betweenness, closeness, stress, and degree to see the most significant or the most important protein which is play as main role on the disease. Okay. From this result, we can see the TNF and EL6 become the most significant protein. IL-6 is interleukin 6 and TNF is tumor necrosis factor. According to Puto et al. On 2014, IL-6 and TNF is a cytokine pro-inflammation, which is managing the production of antibody or immune. Therefore, when there is a disease that is related to this protein, the production of antibodies will increase massively and become inflamed. Because these two proteins play a role as the main problem on arthritis, for improvement on drug discovery, maybe we could focus on finding out compounds that could inhibit the activity of IL-6 and TNF in causing arthritis disease, so that it will minimize discomfort feeling on this disease and help accelerate healing. This is a picture of IL-6 play a role on mediation of chronic inflammation and joint destruction. Because we know that IL-6 is the significant protein, and we know this pathway. So for improvement the drug discovery, maybe we could find out the compound which is inhibits the activity at this one. Maybe by binding the IL-6, so the neutrophil not gets stimulated by IL-6 and become mediation of chronic inflammation. Or we could find compounds which is inhibit at this one. Inhibits to stimulate RANKL to give an effect, osteoclast activation, bone resorption, and become joint destruction. So because we know this protein is significant and the pathway we get it too. So for improvement for drug discovery is to find out compounds which is inhibit at this area. Okay, thank you for your attention. I think that's all.
Yes, thank you.
Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you for group 13. Maybe I have one question. If you, for example, analyze the protein obtained from your analysis, interleukin 6 and TNF, what's actually characteristics of these two protein in their network? Are they also have they also have a hike between the centrality or also decrease centrality and so on? Can you explain the characteristic of these two protein that you obtain from your analysis? Related to the topology approach.
Allow me to answer your question, Mr. Wisnanta.
Yeah.
For the identification from those protein TNF and IL-6, they have a quiet grid point on betweenness, closeness, stress, and degree. And then two of this protein is include of two cluster that we got. So after we do cluster, we get two cluster, and this protein, these two proteins, is include on the cluster 1 and cluster 2 too. And then the score of the 4 parameters is quite high. So that's the.
And the overall parameters also the highest. Height also the highest overall centrality.
For the centrality, for the overall centrality, it's kind of unique because on the cluster 1, IL-6 have highest one is the highest one of overall centrality value, but on the cluster 2, TNF is the highest one. So. Okay. Because two of this is always appear on the two cluster, and two of this protein have a grid value on their own cluster, maybe we can say that. So we pick these.
Important.
Yeah, so we pick these two protein is important, and when we find out the literature about this protein is significantly become a main role as the main role on the disease. Sorry.
Okay, thank you, Mr. Arif, and also the other members of Group 13. Thank you. Now I will ask Group 14, the last group, to present their work about PPI analysis. Please, Group 14, time is yours.
Okay. Hello everyone. We are from 40 group. I'm Momoka Mitsuguchi. Our member is Eliza, Ayu, Ira, Noni. Today we would like to introduce coronary heart disease to you. Firstly, we will tell you a little bit about what coronary heart disease is. Next, we will describe results of the cytoscape and PPI analysis. Finally, we will talk about next plan. So let's begin. Coronary artery disease, CAD, is one of the major cardiovascular diseases affecting the global human population. This disease has been proved to be the major cause of death in both the developed and developing countries. Lifestyle, environmental factors, and genetic factors as risk factors for the development of cardiovascular disease. The risk factors of CAD include diabetes, hypertension, smoking, obesity, and psychosocial stress. The eradication and management of CAD has been established through extensive studies and trials. Anti-platelet agents, beta blockers, and calcium antagonists are some of the few therapeutic agents used for the relief of symptomatic angina associated with CAD. Next, please.
Noni? Oh, okay, maybe I will explain about this. It's a string DB result. After we input the CAGD database into string, we can get a design of interaction of protein in CAGD. Next slide. From the string DB, we get a database knowledge and show that this protein was involved in several pathways, like pathway in cancer, RAGE signaling pathway in diabetic complication, with shear stress and atherosclerosis, then cytokine receptor interaction, cholesterol metabolism, HTLV1 infection, NEPK signaling pathway, proteoglycan in cancer, P1CK AKT signaling pathway, then paratosis. Next. Then TSV from StrainDB was processed in cytoscape and showed and resulted 5 clusters, and this cluster 1 have a minimum p-value, and this is a several protein that interact each other, SEPIN1, F8, and ATC. Then this cluster 1 was processed into overall centrality. Next. This is the resulted database from overall centrality, and it is so the highest closeness centrality and betweenness centrality is APO B and APO A2, and even if from the overall centrality data we get negative, but we make strength with the cytoscape result data. Next, from the first, which is we know that the degree in APO A2 and APO AB was 11 value, and it's the highest value. Next. We know that ApoB is apolipoprotein B10, which is a major protein constituent of kilomicron, ApoB48, LDL, and FLDL. ApoB100 function as a recognizing signal for the cellular binding and internalization of LDL particle by the ApoB receptor apolipoproteins, whereas ApoA2 is apolipoprotein A2 may stabilize HDL structure by its association with lipid and affect the HDL metabolism, apolipoproteins. Next. So we conclude is that the protein in cluster 5 was involved in cholesterol metabolism, in cholesterol pathway. Next. And we find in the, I'm sorry, maybe it's Ayu will explain it. Next, Ayu.
Okay, inhibitor APOB mechanism of action. Mipomersen binds to the messenger RNA coding for apolipoprotein B100, a protein that is the main component of low density lipoprotein and very low density lipoprotein. As a consequence, the RNA is degraded by the enzyme ribonuclease, and APOB 100 is not translated.
Maybe it's so it was one of the application, one drug that use ApoB protein target. So the next plan is we know that the protein target from CSD is ApoB, ApoA2, so we can find natural product ligand. Then we do the molecular docking to find a leading drug for CHD. Next. Okay, maybe it's the presentation that from group 14. Thank you.
Okay, thank you, group 14. I have one question. Maybe the question is actually same that I asked the first group, yeah, the group 8. What actually the advantage and disadvantage that method on this approach?
Yeah, maybe the advantage is tool that make us get a center protein that's involved, that's important in one disease, so we can choose a right protein target that we can imply it to others method like molecular docking, etc., pharmacophore and fatwa screening, and etc. Like,
and what the disadvantage, maybe the drawback of this method?
Or disadvantage.
On the limitation of this method?
Maybe accuracy, or the algorithm, maybe like that.
Yes, because maybe accuracy of algorithm actually depend on the parameters, and we should set the many parameters. So the optimization of parameters has become a great issues. Yes, if we, for example, if we cannot set the optimal parameters, then maybe the result is not so confident or not so accurate. Thank you for group 14. I think all of group already make a presentation, make an excellent and interesting presentation. Thank you for all group from group 8 to group 14. I learned a lot of new knowledge, especially how to interpret the protein or the result, the protein obtained from PPI analysis. In my opinion, I think after I saw this presentation, all of you, all of group have already known how to use PPI analysis to find the protein that have important role in certain, for example, in certain disease, and can use this result to explain the mechanism as a for example, like a biological process or something. So I hope we can do such a collaboration maybe for the future by work together in research or something, because my background is computer science, and many of you, a lot of most of you is in non-computer science, but related to the such domain problem. So I think the collaboration will be very interesting, I think. Thank you, all participants, and I will give the time to